today I'm going to talk about aging in terms of heterogeneity. And in particularly, I want to talk about how different organ or tissues within the same individual appear to age differently. So I guess it is like a common knowledge these days, aging heterogeneity is everywhere. It happens across different scale. You can see different species, they have different lifespan. And even if we look at different organ systems within the same individual, we can see like their aging rate also differs. For example, this figure I put here is come from a review paper. In that review paper, the authors have summarized the aging rate among different organ systems in women. So people have used the physiological measurements to show how different organs feel with age. You can see clearly in women, the reproductive organ system age the fastest. In contrast, the neurological and gastrointestinal system appear to age the slowest. And similarly, if we can use physiological measurements to document aging rate, we can also use molecular data to show aging trajectories. For example, this figure here shows like the authors have used bulk RNA-seq data to profile the gene expression from 4 organs in mice at both young age and old age. And we can see for different like colors, it is very clearly the gene expression profile in different organs. They also follow different aging trajectory. And nowadays, actually, we have a number of so-called aging hallmark to measure aging rate. And one of the aging hallmark for molecular data is the gene expression variability. And this cartoon shows the meaning of gene expression variability. If we took a number of cells from a young individual and also a number of cells from an old individual, and we measure the gene expression profile of certain genes, we can see for gene expression, maybe the mean expression level will not change with age that dramatically, but the variance of gene expression may increase with age. And this pattern was first reported in 2006 when people were still using quantitative PCR, and people have used this early day technique to measure gene expression in heart cells in mouse and saw this pattern. And later, with the rapid development of sequencing technology in 2017, people used the single-cell RNA-seq data technique to show this pattern also existed in pancreatic cells in human. And then more and more large-scale single-cell data came out, and people have documented this pattern in a number of different organ systems and in a number of mammalian species. So when we were reading these papers, we also download their data and perform the analysis to see how global or universal this pattern is, and we notice something different. So the first thing is this pattern is not a universal pattern. So some cell type, their gene expression variability increase with age, while other cell type, their gene expression variability barely change with age. And second, even for those cell types that their gene expression variability increase with age, their magnitude of change also differ a lot among different cell types. So this brought up this question: why does gene expression variability change with age in some cell type but not in others? And a paper gave us some clue, an early paper in 2007. And so in 2007, Warren have this paper in aging cell. And in that paper, he just took a number of blood cell types from mouse in young age and old age. And similarly, he used quantitative PCR to measure gene expression variability. And he saw the gene expression variability barely change with age. And in the discussion part, he mentioned, okay, this might be due to that the blood cell types, they have a fast cell turnover rate. Later, there's a review paper in 2021, which proposed a very similar idea consistent with the 2007 paper. So in this 2021 review paper, Randall and Westcary, they came up with this cartoon, gave us a direct idea of how aging at a single cell level were linked to aging at the organ cell level. So as we can see, different individual cells within the organ, they also have their own aging trajectory. Some cells are pretty young, while other cells are already very old. So in their review paper, they mentioned this idea, like different cells have their own cell age, and this cell age structure in different organ system, they may reflect the system level functional decline that we see at a macro scale. So inspired by all those previous work, and we came up with this cartoon to illustrate our idea. Just as every human individual have a chronological age, every cell also has its own chronological age, or refer to it as a cellular age. So for different organ systems, some of them have a fast turnover rate, while others have a slow turnover rate. If we look at the organ with a slow turnover, the cells, they infrequently replace themselves. So they may age together with the organism, which means at a young age, all the cells are also young, but at old age, a lot of cells are already pretty old. In contrast, for organ system with fast cell turnover, the cells keeps replacing themselves. So even if we look at old age, at all the organisms measured at the animal's age, the cells, they may still be very, very young. So we notice that different types of cells, they have different turnover rates, and this might lead to difference in the cell age distribution among different cell types. So we want to test the hypothesis that cell age can explain the cell type specific change in the gene expression variability as the organism ages. And to test our idea, we first need to measure cell age. It is really difficult to physically or technically measure cell type age. But coming from a population genetic model, the cell age is just like a population biology question. We can use a number of clues to infer the cell age. And one of the key parameters actually is the cellular lifespan. So for cellular lifespan, there is indeed some empirical data. So for example, this paper from 2021, they have summarized a lot of previous experimental studies showing the cellular lifespan and came up with a final table showing how different cell types, their cell lifespan changes within the same organism. Here I show some example. As we can see, indeed, for different cell types within the same individual, their cell lifespan change a lot or vary to a greater extent. So for some cell type like neuron, their lifespan is extremely long. And people always believe the neurons can actually outlive an organism's lifespan. And for other cell types, such as monocytes and epithelial cells in the gut, their cell lifespan is extremely short, only 1 or 2 days. So indeed, the cell lifespan changes a lot among different cell types within the same organism. And with the cell lifespan data and also a set of simplifying assumptions, we first translate these cell lifespan data into cell turnover rate. And then we build a cell demographic model to estimate the cell age distribution. So I will skip all the technical details here, but for the cell age distribution, it's basically we can input the animal's age and the cell turnover rate, and it will give us a cell age distribution. So this is the demonstration of the output of that model. For example, if we look at monocytes in blood, which have extremely short lifespan, it's only like 0.9 days, and has a fast turnover rate. And with the model, with the animal's age at 3 months or 24 months, we can draw the cell age distribution at these two age groups. We can see for the blue curve and the yellow curve, it barely changes. So for fast turnover cell, the cell age distribution, they keep unchanged, even if the animals are aging. If you look at the mean cell age, we can see there's the delta age, or we refer to it as delta age. The delta age equals 0. And if we look at a different cell type, which is like endothelial cells from the liver, have a moderate lifespan, which is 67 days, and a medium turnover rate, we can see now the cell age distribution, it changes with the animal's age. It becomes like older for the older animals. And similarly, if we use the mean cell age to measure difference, we can see the delta age now is bigger than 0. And for the extreme, if we look at neurons, which are aging together with the organism, we can see the cell age just change together with the animal's age. And if we use the delta age to measure this magnitude, we can see it is a pretty big number. So indeed, with the different cell lifespan or cell turnover rate, different cell types in the same organism, their chronological age change with age indeed differ a lot. So previously, we have this cell demographic model, which help us to show how cell age changes among different organ systems. And what about changes in the molecular space? What about changes in their transcriptome? So to measure this, we use this mouse aging cell atlas because it is one of the most comprehensive aging atlas at a single cell level for mammalian species. So for this data set, they have single cell data for 23 tissues and 76 cell types at 3 months or young age for mice, and also 24 months. So because it's single cell data, so we can measure gene expression variability at this cell type level. So we use the metric of CV or coefficient of variation to measure gene expression variability for each individual gene. So similarly, if we look at one gene, and we can calculate this CV for this cell type at this young individual. And when we look at the older individual, we can calculate the respective CV for this older individual. So for every gene, we have two values, CV at young age and CV at old age. And because we have a large number of expressed genes, so if we plot them together, we got this variant plot, which the blue variant represent CV of all expressed genes at a young age, and the yellow shows the counterpart at old age. And we can see there is some trend changing from young age to old age. So we use a linear mixed model, and we extract the coefficient, and we refer to it as the beta age, which shows the change in transcriptional variability with animals' age. So we use this data set and the CV metric, we can calculate beta age for all these cell types in mouse. We can see the beta age is pretty small for monocytes in blood and gets bigger for neurons in the brain. And just like several earlier slides showed, we also have the delta age measured for all these cell types. So now we are ready to address the question, what is relationship of change in transcriptome space and the change in the cellular chronological age? We can just contrast these two metrics to see their relationship. So for each cell type, we have this delta age metric, and for this beta age metric. When we plot one against the other, we got this scatter plot. And surprisingly, we see there is a very, very high correlation between these two metrics. Actually, more than half of the variance in the beta age or change in the transcriptome space, they can be explained by cell age difference reflected by the x-axis. And we were very surprised at that time because we always look for some explanation for why some cell types show bigger changes in transcriptome space while other cell types show like trivial signal. And it looks like the cell turnover plays a major role in this setting. So let's look at this cartoon again, and we have a much clearer story to share. So for different organ system, even if they are in the same organism, they have a very different cell turnover rates. So for some organs which have a slow turnover, their cells age together with the organism's age. So we see a large cell age difference between young and old animal. And at the same time, we see large changes in the transcriptome variability. And for organs with a high turnover rate, because the cells keeps replacing themselves, so the cell age did not change that much, irrespective of the organism age. So the cell age difference is pretty small between young and old, and we also see trivial changes in the transcriptome variability space. So what about the cell types that are the same cell type but reside in different tissues? For example, endothelial cells, we know endothelial cells, they exist in different tissues across the human body. And indeed, in the mouse aging atlas data, there are 12 endothelial cells in different tissues, as you can see, like the points here. And if we don't have tissue-specific lifespan data, or all those endothelial cells only have one lifespan estimate, the data would look like this. So they are all anchored by the same x-axis here, but their gene expression variability may change, differ from tissue to tissue. And lucky for us, indeed, there are some tissue-specific lifespan data there. So there are 4 endothelial cell types which have tissue-specific lifespan estimates. Now we can spread these 4 data points across the x-axis. So one possibility when we do this is these data points may just randomly spread out, which means the cell age is not a big thing here. Or alternatively, if the cell age is indeed a huge parameter, we will see the 4 data points would spread out following the global pattern, which is the cell. And when we plug in the real data of this using this tissue-specific lifespan estimate, we see indeed the data spread out following the global pattern, which is the cell. So our explanation is the tissue microenvironment influence the tissue-specific lifespan turnover rates. And this in turn influence the cell age of those same cell type but residing in different tissues. And the cell age is a key parameter determining the amount of changes in the transcriptome space. So that's why we can see this pattern in the real data. And from previous analysis, it looks like the long-lived cells, they just accumulate a lot of changes in their gene expression space, which is pretty counterintuitive because we know like the neurons, although they are long-lived, but they don't seem to experience rapid functional decline in the animal's aging trajectory. And we know like the cells, they can accumulate various kinds of molecular damage, such as misfolding of proteins, mitochondrial dysfunction, and also DNA damage. But at the same time, the cells also have a lot of strategies or molecular pathways coping with these damages. For example, they have chaperones which can correct the misfolding of those proteins. And they have a lot of other metabolic pathways which can correct the mitochondrial dysfunction and also DNA repair system. So we wonder, given we see a lot of changes in the transcriptome space, and we also know the cells can activate molecular pathways to cope with molecular changes, are there any gene functional groups whose expression change also correlate with cellular lifespan? So given that hypothesis, we do some functionally related analysis. So we use the geoterms from the gene ontology database, and we just calculate the gene expression variability for each geoterm. Here shows several examples, and because it's single cell data, we can calculate this at this cell type level. So we calculate the gene expression variability for different cell types in these geoterms. And we also have the cell lifespan data, so we can address the question. So what is the relationship between cell lifespan and these functional gene group activities? So this figure shows the broad pattern. So on the x-axis, they show the cell lifespan of different cell types. And for the y-axis, in each panel, it shows the gene expression variability of different functional gene groups. And we observe very interesting patterns. So for protein folding related geoterms like chevron-mediated protein complex assembly, chevron-mediated protein folding, we see a positive relationship, which means the cell types which have a long lifespan, they invest a lot of their gene expression resource in maintaining their protein folding homeostasis. And in contrast, we see for mitochondrial DNA repair and DNA synthesis involved in DNA repair, these geoterms, they show a negative relationship with the cellular lifespan, which shows like for fast turnover cells, they actually invested a lot of energy and resource in maintaining their DNA repair or DNA complete system. And this pattern gives us this idea that different cell types, given their lifespan, they actually use very different molecular strategy coping with cellular age accumulated molecular damages in each individual cell. And given like the protein folding is a really big functional requirement of cells, and we just saw like the long-lived cells, they activate more genes involved in these protein folding pathways. We wonder, do long-lived cells, they also prefer to express genes which have a more stable protein? For different genes, their encoded protein have different inherent stability. Some genes, their translated amino acid sequence are inherently more stable. So we wonder, do long-lived cells, their expressed gene profile, their encoded proteins are inherently more stable? And for protein stability, like you need to use a lot of physiological measurements to measure how stable it is. But in previous study from an evolutionary perspective, people have observed this pattern that more stable proteins are associated with more higher evolutionary constraint and lower evolutionary rates. So instead of measuring protein stability, which is pretty troublesome and remains a lot of experimental work, we can use evolutionary index to approximate these protein stability and then check the relationship between cellular lifespan and the expressed genes protein stability. So we use DNDS value from a population genetics area, which is highly related to evolutionary constraint. So similarly as a functional gene group analysis, for each cell type, we have a cell lifespan data, and we also measure their evolutionary rate for each cell type. And for this figure, each dot represents one cell type, and we do this analysis for young animal and old animal separately. And we can see in both age groups, there is this negative relationship, which shows like for cells which have a longer lifespan, their expressed genes have a lower evolutionary rate, which means they have gone through higher evolutionary negative selection, and their proteins are likely to be more stable. So this reinforced the idea that long-lived cells, indeed, they experience a lot of proteostatic pressure, and they have activated their molecular pathways to keep their protein stable, and they also prefer to express genes which with a highly inherent stability. So a summary of all the analysis shown in previous slides, we were intrigued by this question, like why different cells, they age differently in their gene expression space with animals' age. And we have identified this K parameter, cellular lifespan, and we have examined a number of different evolutionary and functional properties associated with this lifespan. And we see very, very interesting associations. So all those associations prompt us to give this heuristic model explaining how individual aging at a single cell level linked to the organ level. So if we look at the cellular aging at the individual cell level, we can see at the cell ages, different molecular damage accumulate inside the cell, such as misfolded protein, DNA damage, epigenetic drift, and mitochondrial dysfunction. But at the same time, the cell actually have a lot of strategies coping with these damages. They can activate DNA repair, epigenetic maintenance system, mitochondrial maintenance system, and also proteostasis to keep the cell in check. And when the damage is reach a certain amount, the cell have to go through the cell death or cell senescence process. And this is the broad picture for individual cell. And if we change our view from this aging pattern at a single cell level to this organ level, we can see like for organ or tissue system, which have a very, very slow turnover, the cells age together with the animal's age. And to cope with all those accumulated damages, the cell indeed activate a lot of pathways to clear those damages. And this is a strategy for cells in slow turnover cells, but clearly not a strategy for cells in fast turnover cells. Because for fast turnover cells, these cells, they can avoid the cost of maintaining homeostasis by simply being replaced. So for these cells, when their cellular damage accumulated to a certain level, they can just go through the apoptosis process, and the stem cell in those cell organ system can give rise to new cells which complement the loss of cells. So we speculate actually in these fast turnover cells and in cells with a fast turnover rate, their damage accumulation rate may go hand in hand together with their cellular lifespan. And this is our heuristic model trying to link aging at a single cell level at a broader organ level. And we tested this model previously using the single cell data in the mouse aging atlas. And nowadays we have a lot of spatial data. So currently we are extending this work to the spatial single cell data and to see if the cell turnover rate is still a big predictor in different spatial single cell data across the different organ systems. So that's all for the research part. And because most of the work were done when I was a postdoc in University of Washington, so I want to express my deepest gratitude to all the support and discussion to the lab from Daniel Promised lab in University of Washington. And that's all for my presentation. And thank you for your attention.